So I thought I'd start by introducing myself and just briefly talking about how the projects that we're doing in MedArc started. So I'm a postdoc at the Princeton Neuroscience Institute. I started my postdoc in the summer of last year, and I was brought on to try to do real-time neuroimaging with fMRI, specifically trying to eventually do real-time reconstructions. And so when I first started my postdoc, I was kind of not that good at machine learning, I'd say. I wanted to expand my skill set, so I went looking for resources for how to learn implementing neural networks and stuff like that. And that made me join Lion, the Discord server. And when I joined Lion, I found a channel called Medical General. And I joined that because that seemed up my alley. And I saw that they were already trying to do reconstructions of images from fMRI brain activity. So that was obviously very interesting, and I looked into what was going on and saw that not much progress had been done. Tanish was leading this project using a sort of outdated data set, and so me and Tanish got talking, and I basically started leading this reconstruction project using the newest data set available, given that I had more fMRI experience and the community had a lot more machine learning experience. So that worked out well. And eventually Tinch started MedArc and invited me to hop on board to start various projects related to both this reconstruction project and various other projects that are now going on actively in the Discord server, and which are if you're not familiar with the MedArc server, but the fMRI general channel is like a general hub for the different projects that are going on with neuroimaging. And then there's different working groups for more specific projects. And so a really interesting background of this paper is that all of it was worked on openly with volunteers, both the Princeton team, but primarily the MedArc team, working just publicly on GitHub and using compute from Stability that they generously shared with us. And so over the course of a little less than a year, we were able to get this preprint done and submit it to conferences and then show the preprint online on Twitter. So that's a bit of the background. And we have a project page here if you're interested to learn more. I can briefly show that. So if you haven't read the preprint or you want to look at the code, then there's a project page that shows all this off. So that's kind of a background. I'll go through these slides, introduce you to how MindEye works and what goes on with that. Feel free to interrupt me at any point or save questions for the end if you prefer. You can also chat in the chat box, although I probably won't read that while I'm presenting. All right, so here's some examples at the bottom of this title slide. You can see that there's a scene image, that this is an image that a person is looking at inside of an MRI machine. And then the goal is, of course, you're recording their brain activity while they look at the image, and you try to reconstruct what the image was from the brain activity. Here's a kind of time course of progress in this since 2011 with a groundbreaking study from Nishimoto. Groundbreaking at the time. This is actually a video clip, so it was even harder than what we were doing, which is static images. But you can see that roughly you can get maybe the color of an object, but things have really improved over time. And I think a lot of the improvements have to do with a much better data set being available, which I'll talk about on the next slide, and also the release of really high quality pre-trained multimodal models like CLIP and generative models like Stable Diffusion that we end up using to train MindEye. And I also want to emphasize here that reconstructions are not meant to just be returning the original image that's being seen in the scanner. So it's not just a novelty kind of project. There's real important theoretical and clinical kinds of work that can be done if you have a high quality reconstruction model. And that's because you're reconstructing the sort of internal mental representation of what is being perceived. It's not just like hooking up a model to your retina. Like it's not as informative. It's like you're actually being able to look at different parts of the brain that are involved in different functional specializations. If you have neurological disorders or attentional biases or individual prior experiences that would change how you perceive or attend to something, then all of that will influence the subsequent reconstruction. So there's a lot of different avenues for research that can be done if you're able to reconstruct images from the brain. So this is kind of a general purpose tool, it's the kind of idea behind this. So this is the natural scenes data set. We did not collect this. This is a different group that put out this very high quality data set that we are lucky enough to be able to use. So this is a data set of 8 subjects who were each being scanned inside of a 7 Tesla MRI. Each of them underwent dozens of scanning sessions. Excuse me. Okay, so there are 8 subjects. Each underwent 30 to 40 hours of scanning, looking at individual images one at a time. Sorry, I got back from a conference this week and my throat's kind of shot. So please deal with me. So there's a person in a scanner. They're lying down on a bed, looking at a mirror. The mirror is getting projected from a computer screen showing natural scenes one at a time. And there's 30 to 40 hours for every person looking at these images. And the data is pre-processed in as high quality as you can offline. And so I want to emphasize that these reconstructions that we're doing are not in real time. This is data that has already been collected and pre-processed that we are then using to train and then reconstruct the images from a held out subset of the data. Also emphasizing here that these participants willingly had their brains scanned and their data shared. Okay, so I'm sure that you're all familiar with text-to-image generative models like Stable Diffusion. These models get trained on massive data sets, scraping the entire World Wide Web. And with fMRI, even though we have quite a large sample of dozens of hours of scanning, it's still not that big of a data set compared to training like text-to-image models. So the goal is to map the brain data to an embedding space that can then be fed through a pre-trained model. So in our case, we map to CLIP, which is a multimodal embedding space trained between text embeddings and image embeddings. And so once we're able to map to CLIP image space specifically, then you can just feed that embedding directly into a model like Stable Diffusion to output a realistic looking image. So I'll talk a little bit about what CLIP is here. But again, this is just, you can use this approach to map to any multimodal space. We happen to use CLIP because that's something that's, you can use that embedding space to feed into other models. CLIP learns to match images and their text descriptions together. So for instance, here, there's a stack of n images and n captions. So image 1 might be a dog, text 1 might be Pepper the Aussie pup. And so given this batch of image text pairs, you can have a matrix like this. And the idea is that every caption gets encoded to an embedding space, every image gets encoded into an embedding space, and these models are transformers. And the model tries to maximize the cosine similarity of the diagonal here, the positive pair mates, and minimize the off diagonal. And in doing so, is basically learning to represent the similarity between images and their corresponding text. So more specifically, every image in the case of clip, the last embedding, the last layer of clip gives you a 1/768 embedding. You get the same size embedding from text, and you compute the cosine similarity for every single cell here, normalize it, and optimize with cross entropy loss. And with these different generative models like Stable Diffusion, some of them can accept clip image embeddings instead of text. So with regular Stable Diffusion, it only accepts text input. And so it would not allow us to feed in a clip image embedding. But there are variants such as Stable Diffusion image variations that Justin Pinkney trained from Lambda Labs. And this is basically a fine-tuned version where instead of feeding in a text prompt, you can feed in a clip image embedding, and then it will give you a variation of that image. You can see an example of that here with this image. You take the clip embedding, feed it through Stable Diffusion image variations, and it gives you a somewhat similar image. There's also a model called versatile diffusion that can do pretty much the same. And one important difference is that instead of taking the last layer, it takes the last hidden layer from the attention part of the transformer. So instead of 1/768, it's 257/768. And that gets fed through the network and gives you an image variation. And from testing this out, versatile diffusion seems to give much more high fidelity image variations compared to the original input image, probably because there's a lot more signal contained in the last hidden layer. So just to summarize, if you can map voxels to clip space, you can essentially feed that through any of these generative models that accept clip image inputs, like versatile diffusion. So some other papers have already tried to kind of do this approach. So Lin et al., the Mind Reader paper, used convolutions and residual blocks to map to the last layer of clip. Oslick 2023 used ridge regression to the clip last hidden layer. And you can see the results here on the left. Our approach was to use an MLP and contrastive loss to train our mapping. So I'll go into a bit more detail of that. It's essentially a giant multi-layer perceptron with residual connections. So we take a portion of the brain that's most receptive to visual inputs. In this case, 15,000 voxels. These are 1.8 millimeter cubic cubes of cortex that each have a corresponding brain activity associated with it for every image. And so these voxels get linearly mapped and given an activation function to map to a 4,000 by 4,000 vector. And then this undergoes 4 residual blocks. By that, I just mean this for loop here, where it goes through the MLP and you add the residual from the last output into it. And so that goes through 4 residual blocks, and then there's a final mapping. And this gives us the output of 257 by 768, which can then be unflattened and fed through like a generative model. So it's a very simple architecture, very simple mapping, but it is quite dense at 940 million trainable parameters. But this does fit on a single A100 GPU. So it's not that hard to train. So talking about this contrastive learning setup, we also use data augmentation. So this is augmenting the voxels specifically that are input. Data augmentation is like creating fake data so that you have more data to fit your model. So we use mix-up augmentation. Specifically, you generate a random weight from a beta distribution, randomly select half the training samples, and you can replace the training samples with a weighted average with some other sample. This means that the training samples, half are untouched and the other half are slightly weighted towards another sample. And you can see that weights matrix here, where the diagonal is basically untouched voxels, and the off diagonal is being weighted by some other random sample. So we use a variance of mix-up that works with contrastive loss and can be bidirectional. This was devised by Atmadeep, who's the co-first author on this paper. And basically how that works is for every clip image, aka for each row, you multiply the vector of log probabilities by the weights matrix that you got from the last slide, take the sum, average across the sums of samples multiplied by -1. It's basically just cross entropy loss. And to make it bidirectional, you repeat the same process for the columns and take the average of the losses. This is basically the same thing as Mixco, cited at the bottom of the slide, but with it being bidirectional. We stop doing this data augmentation after 1/3 of the training, and this is because prior work has shown that it's better to stop doing data augmentation for mix-up after a certain point. And if we stop doing data augmentation, we're able to then switch to a soft loss, basically using knowledge of clip space itself to inform the contrastive training here. The basic idea behind this is that the weights matrix gets replaced by the matrix of clip to clip itself. And so essentially, if you're doing poorly, poor performance on dissimilar embeddings should be weighted more compared to poor performance on very similar embeddings. And this is basically knowledge distillation. So knowledge distillation being that you're transferring the knowledge of clip, essentially. So if the 2 images are already very similar in clip space, then it's reasonable that you're going to have more difficulty picking out which of those 2 is the original one, for instance. So all I've been talking about right now is basically introducing fMRI as a third modality, essentially, into clip space. So clip has text, image, and now there's fMRI, brain. And we train that mapping in the same way that clip was originally trained, basically. So now we've got this third modality in clip space representing brains. What can we do with it? So the 2 things that we wanted to do are retrieval and reconstruction. So retrieval means how well can you retrieve a corresponding positive pair mate. So for instance, image retrieval would be how well can you find the given image if you input a brain sample. fMRI retrieval or brain retrieval would be how well can you find the corresponding brain sample given the image. So essentially how this works is you're given input a brain sample, calculate the cosine similarity with a pool of candidate images, and whatever has the highest cosine similarity you pick as the presumed original image, and then you see how well that does. With reconstruction, you're actually trying to generate the original pixel image. So this is feeding the clip embedding into the generative model that I was talking about before, like feeding in through versatile diffusion to output an image. And then we can calculate image metrics on that to see how well it's doing. So at this point, you run into a problem in the sense that retrieval works, but reconstruction doesn't work. And that's because you can't just simply input a clip embedding as is through the versatile diffusion or stable diffusion models, because these clip embeddings have been trained to maximize cosine similarity, essentially, not to be interchangeable exactly one for one with the corresponding positive pair mate. So in other words, they're aligned in terms of like the vector direction, but they're not like substitutable in terms of magnitude. And this is the same problem that happened for DALL-E 2, essentially. So if I'm visualizing here like clip text embeddings, clip image embeddings, and you can see using dimensionality reduction that the manifolds are basically, they're in totally different spaces and therefore not substitutable with each other. And you get the same thing between clip fMRI embeddings and clip image embeddings. And so the thing is, we need this green cloud of fMRI embeddings to be basically overlapping and substitutable with the blue cloud. And right now they're totally separate. So DALL-E 2 had the same issue, and you can see this figure here as this is how they addressed it, and we basically copied them to correct this problem. So DALL-E 2 addressed the disjointed embeddings problem by using a diffusion model. So they have two diffusion models, right? They have a diffusion model that goes from clip text and transforms the clip text into clip image, and then the clip image goes through a second diffusion model that gives you an actual image at the end, pixel image. So this first diffusion model that goes from one clip embedding space to another clip embedding space, they call a diffusion prior. And so there's some open source code that implements this diffusion prior, which we are very thankful for. And we essentially used and modified this code base to implement our own diffusion prior. Instead of going from clip text to clip image, we go from clip fMRI to clip image. And so that changes the schematic of what's going on here. And so this is the updated schematic for the MindEye pipeline now that we have this diffusion prior. And so I'll walk you through this. We have image brain pairs. So every image has a corresponding activation, flattened activation, spatial patterns of 15,000 voxels. These 15,000 voxels get mapped through an MLP backbone, which is what I showed on the prior slide with the residual blocks. And that maps it into a 257 by 768 space. Then this gets split into two different submodules, one specialized for retrieval and one specialized for reconstructions. So for retrieval, all it is a final feed forward network that takes the last dimension and maps that through linear projection. And then that gets fed through the same contrastive loss that I was talking about in the prior slides. For the reconstruction submodule, the backbone outputs go through a diffusion prior that we trained from scratch, mapping from 257 by 768 to, again, 257 by 768, but it's trying to actually minimize the mean squared error between the two spaces. So actually like being substitutable with each other, in other words. So for retrieval, we don't care about them being substitutable because we just care about the directions being consistent. And we found that if you try to minimize for both these objectives at the same time, then they kind of compete with one another. So you have to separate them into different submodules in order to be able to accomplish both of these tasks at the same time in a single model trained end-to-end. So the outputs of the diffusion prior we're calling the aligned clip fMRI embeddings, and then that can get fed through the versatile diffusion model to give you a reconstruction like this. So now I'm visualizing the UMAP dimensionality reduction for the different parts of that schematic on the previous slide. So you can see now that everything gets trained end to end, things look a bit different, but the MLP backbone is obviously still disjointed, and the outputs of the MLP projector and the diffusion prior are more or less overlapping, especially so for the diffusion prior, which is what we are specifically trying to get to work for that. And as a side effect, we found that it also improved the retrieval performance at the same time. So it was a win-win. So now let's get to the actual results. And for retrieval first, I'm going to walk you through this. So we have these voxels that get mapped to the disjointed clip fMRI space. And we can basically have a pool of images, image candidates, calculate the cosine similarity between every image and the MindEye outputs. Whatever has the highest clip similarity, we pick as the predicted original image, and then we see how well that does. This is the top one performance of finding the exact original image. And with subject one, given a test set of 982 images, it gets 93.2% accuracy, which is really, really high. Like compared to other papers, especially given the fact that a lot of these test images look very similar to each other. So when I looked through the test data set, these are images, by the way, that were never shown, none of these images were included in the training set. So like all of these zebras, none of the brain activity associated with looking at these zebras was used to train the model. So in the test data set, there were like 14 zebras. There's a lot of similar looking images, I guess, as a side effect of using COCO. And so even in the case of 14 zebras, it can reliably find the right one given all of these candidates, which I think is really great and shows off the fine-grained information that the MindEye embeddings have, which is really important because I think a lot of other reconstruction kind of pipelines, they might be able to tell you that it's a zebra, but it might just have the high level category and lack some of the more specific information of like, can it really tell the difference between very confusable images? Another cool thing about this is that, and this is novel to MindEye, to my knowledge, trying to link to a large-scale image database like Lion 5B. So there's billions of images in the Lion dataset. You can compute the cosine similarities for all of the billions of images and compare that with the MindEye outputs for a given image. And we find that if you do this, it can work. So obviously, you're not going to be able to retrieve the original image, assuming it's not in the data set itself, but you'll be able to get very similar looking images without a reconstruction generative model at all. So these are not generated images. These are like, static photos that were scraped online by the Lion team. So, looking at a tennis court, it can find a similar looking tennis court. So, if you're interested in not using generative models, which might be computationally difficult, this is another way to do it, and seems to work pretty well. So for reconstruction, there was one thing I left out so far, and that's that we actually have a separate independent low-level pipeline. So how this works is that we map the voxels through a similar kind of MLP as what I described before, but this time mapping to the stable diffusion's variational autoencoder embeddings. So this maps to a 64 by 16 by 16 embedding space and then gets upsampled with convolutional layers, and then minimize the mean squared error for that, and also use contrastive loss, this time not with clip, but with a embedding space that's specialized for low-level tasks like linear segmentation. And so the idea here is that as long as we get some sort of blurry, perceptually similar starting point, we can improve the reconstruction. So you can see here that if you adopt this approach, you don't really get a surfer, but you do kind of get a blob where the surfer is, and maybe the colors are somewhat similar. And compared to starting from pure noise, which is what would happen if you didn't have this pipeline at all, this is a much more informative starting point. And we find that this is really great for increasing the lower level image properties, like lower level perceptual things like object location and color in these reconstructions. So to be more specific with image to image, the denoising diffusion process is you noisify the low level reconstruction, use that as the initialization for the diffusion instead of starting with pure noise. And so it will take on some of the qualities of this in creating the reconstruction. So here's some reconstructions from MindEye for subject 1. This is, remember that there's 3 instances, there's 3 same image repetitions for every unique image. So we have data of subject 1 looking at this bear 3 times. All 3 of those times are in the test, the held out test set. And so we average across the 3 samples and then feed that into the entire MindEye pipeline from the previous slide. That reconstructs an image, and that's what I'm depicting in this plot. And so these are some of the best looking ones. There's some variability here. All 982 reconstructions are on the project page website if you're interested in looking across all of the different samples. But for these samples, you can see it does quite well in retaining both the low level and high level image properties of the original images. This is what the reconstructions for the same images look like if you only include a single sample. So if you only have 3 seconds of the person looking at the bear, so a single sample, what do the reconstructions look like? So you can see that it does do worse, but still retains some signal there. So it could be adapted to, say, single trial level kinds of pipelines. So this is comparing to other fMRI-to-Image papers, such as the previous best performing paper from Furkan Urslik. And this is a table showing different evaluations for the same test set. And not all the papers reported all of the evaluations, which is why there's some missing data here. But overall, MindEye performs, generally speaking, better than all the other ones, except for structural similarity score. But yeah, it does very well, especially in retrieval. This table also shows the results for if you separate out the different parts of the pipeline. So if you only evaluate the low level pipeline versus high level versus the outputs that you get from Lion retrieval. And this is comparing the low level outputs from Urslik's paper and MindEye. So Urslik also used a high level and low level pipeline to do the reconstructions. And so this is a direct comparison between subject 1's only low level outputs between the 2 papers, showing that MindEye does do better specifically for pixel correlation and structural similarity, which is the lowest of the low level metrics, showing that it does seem to help out quite a lot with retaining like the raw kind of perceptual image metrics. And just to orient you here, the low level is like those color, object location, texture kinds of details. High level refers to meaning-based evaluations, like does it look like a bear if the original image was a bear? That kind of stuff. The retrieval scores are referring to top one accuracy given a sample of 300 random image candidates. So how often can you find the exact original image out of the 300 in the batch? Finally, we have some ablations. So this is retraining MindEye with some parts removed to show the contributions of those parts of the model. So the bottom row here is the full MindEye pipeline, and the others are different parts of the pipeline removed. So if you only have the MLP backbone, it does the worst, which makes sense. If you include the projector portion of the submodule for retrieval, then you can see the retrieval scores go up quite a lot. If you add in the prior, the diffusion prior, then scores get even better. And overall, we use this bi-mix co-training of switching the loss after 1/3 of the way, and that does the best if you're trying to maximize for both the retrieval and the reconstruction at the same time. These are some example reconstructions for those ablations. This table shows ablations for changing the number of residual blocks in the MLP backbone or mapping to only the last layer of clip rather than the last hidden layer of clip, meaning that the number of parameters goes down a lot. So basically, this is showing that we are justified in using a giant dense MLP with these residual blocks because it does do the best. And it's not like we're overfitting with having too many parameters. It seems like that does benefit the performance. And this ablation table is checking out different loss functions. So we, again, use this bi-mix-co novel variant. 1/3 of the way through, we switch to the soft loss. And that seems to do the best across both of these. But the most basic contrastive objective is info NCE, but this is a unidirectional loss, and so it doesn't do well on one of the two retrieval metrics. And given that we can obtain clip image embeddings from the brain, you can feed this into different models. So it doesn't have to be versatile diffusion, as was used in the previous slides showing the best results. You can instead map to the inputs of different generative models. So this is the model that we first tried to map to before versatile diffusions. This is the stable diffusion image variations model where you map to the last layer of clip, and you can see that it doesn't do quite as well, which makes sense because there's less signal in the last layer of clip, and that probably explains why there is a slight performance worsening. There's also this Lafite model. This is actually a GAN, a general adversarial network, not a diffusion model. And this is the model that was used by the Lin et al. Mind Reader paper. And so this is a really good comparison with that paper because we're using the same output model to get the reconstructions. The difference is how we map to the clip space. So it's kind of holding that part of the comparison constant. And you can see here that in general, it does still do quite a good job, but not as good as the versatile diffusion model, which did the best. So to summarize the conclusions of this, MindEye uses specialized submodules for retrieval using contrastive learning and reconstruction using diffusion prior to give us a single model trained end-to-end that is state-of-the-art and accomplishes both of these objectives, even though they're actually kind of trading off with each other typically. we find that mapping to a deep MLP, 940 million parameters, did not overfit the data and instead benefits performance. Using a novel bidirectional version of mix-up contrastive data augmentation seemed to perform the best, especially in this low sample setting. State-of-the-art reconstructions are obtained also specifically for low-level image metrics by mapping to Stable Diffusion's versatile variational autoencoder. Image retrieval does particularly well in the sense that it can find the exact original image even among highly similar candidates, suggesting fine-grained information in the brain embeddings that you get out of MindEye. And you can scale that up to large-scale data sets, such as Lion5B, to allow you to output images from brain activity even without a diffusion model or a generative model at all. You can just pick out from a dictionary of billions of images, basically. So yeah, that's it. If you want more information about the preprint, you can check out the project page here. You can join us as a volunteer to help out with other active neuroimaging projects in medRC if you're interested. We meet on Mondays at 11 a.m. Eastern time. You can also check out the fMRI general channel. I think it's a very unique kind of working situation where basically like anyone who has free time and experience to share can cooperate between like the Princeton team, medRC, and Stability's compute to work together on these really cool problems that are you can get your name on a paper and contribute to these things even if you're not affiliated with a traditional lab or research group. So it could be I think it's a very unique situation where we have basically the ability to collaborate with international volunteers across the world with different experiences, different skill sets. Also, big shout out to Tanish for creating medRC, of course. This has been a great community to work with. And Stability AI goes without saying, we value having the compute to train these models. And also highlight here all of the people that directly contributed to this project. Atmadeep, co-first author. Jimmy did tons of stuff with the GitHub and was involved for the majority of the work on this. Stepan basically conceived of the idea of doing Lion 5 billion image retrieval. Alex helped with mapping to the Lafayette model. And we have a contribution section for all of the other authors, but needless to say, everyone was very valuable in working together. Ken is my PI advisor who was very helpful in supervising the project, and Tanish obviously allowed all this to happen. So thanks to everybody involved, and happy to talk about this project, talk about medRC, anything anyone wants to talk about.
What are you think the next steps to follow up from this paper?
Yeah, so there's a lot of next steps. One obvious one is to do mental imagery. So that would be really cool. So the natural scenes data set did collect data of people imagining the images instead of just looking at them, but they haven't released it yet because I think they want to publish their own paper first on that, but they said that they plan to release it soon. So, I mean, the theoretical idea is that when you're looking, previous research has shown that if you mentalize looking at an image, the similar patterns of activation are observed as if you're actually looking at it. So it would be interesting to see if we can directly just plug in this trained model for the same subject and get it to work with visual imagery. Another obvious way to go is to try to do the opposite of reconstruction. So try to predict the brain given the image rather than what this is, which is reconstruct the image given the brain. And that's what the Algonauts Challenge working group is doing right now.
Thanks for the talk. Can you hear me all right?
Mm-hmm.
Was wondering if it seems like the data that you use is just from the visual cortex. Did you try using like the whole brain?
Yes, and actually, it's more than just the visual cortex. It's a brain region that was outlined as NSD general by the authors of the data set, and they basically calculated how receptive was every voxel in the brain to visual images. And if it was above a certain threshold, it was included in the brain region. So it includes like not just visual cortex, but a lot of the like dorsal and ventral streams, so like inferotemporal cortex too. So it's not whole brain, but it is like a good portion of the brain. We did try mapping to the whole brain, and we also tried mapping to not flattening the voxels. So like doing 3D modeling to try and preserve that spatial relationship. And basically what we found is that the results were worse. And I don't really know why they're worse. Maybe it's just harder to map if you include a lot of extra noisy data into it. Like probably a lot of the voxels are pure noise. There might be some signal in there, but just from trying it out, it didn't seem to improve things. But that's like theoretically, I would imagine that there's a way to use the whole brain and get better data. Maybe a different mapping approach or something.
Hey Paul, do you have this intuition for which images did well and which images were perhaps harder?
Yeah, great question. So it kind of depends on what you mean by do well. So like if you're talking specifically about the reconstructions, the kind of bottleneck in performance was kind of related to how well the model itself can like output the images from the embedding. So not really related to the brain, but related to like how well can versatile diffusion take a clip embedding of an image and reconstruct the like a variant of that image from the clip embedding. So maybe a good example of this is like this example of the chefs. So with like two chefs in the picture, it's able to give you back two chefs, but you can see that there's some variation in how well this reconstruction from the original clip embedding matches the original image. And from when I was testing things out, it seemed like the more complex, like the more objects, especially humans, were in the original image, the harder a time it had with reconstructing images that were relevant to it. I suppose it also makes sense from a brain perspective of like, if you have more stuff in the image, there's more stuff to pay attention to, and maybe that's kind of like makes it harder. If you have like a green background with a single bear in it, then there's not as much stuff to encode or to process in the higher level visual processing areas. But if you have a complex market scene like this, like how are you going to reconstruct every single person in that image? It becomes essentially just a blob of humans with food, generally speaking, on the left. So simple images, single objects, those seem to do the best.
Question on this slide. Was there any analysis done on images that have text in them, and was the text properly reconstructed as well? Like the bus in the slide.
To my, no, the images were generally speaking devoid of text. So the majority of images did not have text in them. Some images happen to have text in them, like the tennis, like you can see EGO in the background. the bus has some text on it, but yeah, it does not seem to be able to preserve the text in the images, which makes sense because it's a lot harder to be able to reconstruct not just the objects and the general like colors and locations and semantic properties, but the specifics of text. I mean, that's hard to do even for text to image models without the brain, right? Like
Right.
they had to develop that to be able to handle text. So yeah, it doesn't really do well with text. And to be clear, this model is only going to be able to work right now with natural scenes because that's the kind of image distribution that was used to train the model. So if you try to give it images that are not in that kind of distribution of natural looking scenes that you would find in the COCO image set, then it's not going to be able to do a very good job.
Do you know
And
that for sure, or like, do you have examples where you tried?
No, we don't have examples because we don't have access to the full data set of like we only have the training samples, and these models are not generalizable to new people. Like you train it on subject one and you test it on subject one, train it separately on subject two, test it on subject two. So we can't really test that, but it's just kind of a thing that I would assume essentially like it's not going to be able to reconstruct out of image distribution as well. And we can test that though, because like I said, they're going to release the imagery data set soon. There's also a synthetic images data set that they're releasing. So I think that was looking at like geometric colored shapes or something. So that would be out of distribution and that would be something to test. That would be interesting to see how well it transfers.
and is there any information available about the dataset itself? Like the different parameters of the data. Like one that comes to my mind is the colors that different people perceive. If somebody is colorblind or perceives some colors differently, how were the images? How was that taken into factor while collecting the data?
Correct me if I'm misunderstanding, but like, are you talking about like individual subject specific kind of
Yeah,
differences?
yeah.
Yeah. Like, so if a person were colorblind, which they were not in the study, but if they were, that would change things quite a bit. So I don't know exactly how that would affect things, but like there would not be different kinds of signals that you can train the model on based on different colors. So I would imagine that the colors that get output from the reconstructions, it would not really be able to pick the right color. It might do well just as a side effect of learning from clip space itself. So like it might know it's a tennis court, and then because it's kind of learning from clip as a teacher, it would be able to get green grass instead of red grass, just because it's kind of out of distribution of what it's been taught from the embedding space. And that would not be indicative of the subject actually perceiving the color correctly. That would be kind of like an interpretational issue of using a teacher that is malinformed. So you would have to like account for that. But other things like maybe you have like object agnosia or something, like you're not able to attend to certain aspects of the images, that would probably be preserved in some capacity, like objects would not be reconstructed as well. And even in the case of color perception, like you might get the correct color green, but compared to a normal person, you're not going to have the color metrics be as high performance. So it would be a difference of like comparing to controls versus actually looking at the reconstruction and using that to meaningfully understand what was being perceived by the person, if that makes sense.
Got it. Got it.
There was
So
also
is there
an interesting, oh, sorry to cut you off. I'll just mention there was a cool study that came out today from that I posted in the fMRI general chat, but they reconstructed visual illusions. And so people looking at visual illusions in the scanner, and they found that the illusory percept was reconstructed. So they perceived something that's not actually presented on the screen, and that was shown in the reconstructions. But I believe that they had to use a GAN instead of a diffusion model to get that because of the issue I just talked about, where diffusion models are very informed by the statistics of like what's being used.
It's actually really interesting. I'm going to look that up. Never thought I'd hear GAN again.
Yeah, there's some usefulness to that.
You mentioned that the model is subject specific. Have you tried training on all the subjects and then fine tuning on each one?
Yes, we haven't done it, but that is on the agenda, and we need people in the group to try and help us with this because we have a lot of things we want to do. One of which is across-subject modeling, and this is something that we talked about that I think is currently the. Let me bring this up here. So we have different like working groups, and the active learning working group, we had some discussion about this where we wanted to map basically all the subjects into the same space and then do the approach and see how well that worked. Didn't try it, but definitely something to try. And there's a couple other approaches that are in the same vein of like generalizing the model across subjects. This is related to a different working group of training a foundation model to create a new embedding space generalizable across subjects. So, I mean, if you want to apply fMRI-to-image reconstructions to meaningful contexts like clinical domains or new theoretical experiments, you're going to have to solve the issue of these models generalizing across people, because like I said, the subject that was responsible for allowing us to reconstruct these kind of high quality images, they spent 40 hours in the MRI machine. I mean, that's insane. And if you only have a couple of hours, you do not get reconstructions that look anywhere near as good as this. So it's like $1,000 an hour to scan. So it's just infeasible for most real world applications at this point to be able to do these reconstructions unless we solve that issue of across subject modeling.
You mentioned that you came in with like a fMRI background as opposed to a more ML background. I'm curious, like, what kinds of things did you bring or like understand about the problem that maybe a naive machine learning person didn't understand?
So I think it was helpful to have an fMRI background going into this because there are aspects of fMRI specifically where you have to realize how noisy the data really is and how data contamination works. So one thing that may not be obvious to most people is, so the person's looking at an image for 3 seconds, right? Then there's a blank period of looking at nothing for 1 second. And then a new image gets shown for 3 seconds. And then 1 second blank, 3 seconds new image, right? fMRI activity is a measure of blood oxygenation, where the parts of the brain that are most involved in processing the image, like maybe there's a part of the brain that's like patterns of voxels that are very much care about giraffes. And so they expend more energy. It needs more blood, more oxygen to flow to that area of the brain to recoup the cost of processing that giraffe. That process of bringing the blood in, there's a hemodynamic response function where it takes like 8 seconds for the peak of that activation to occur. And the fMRI is measuring the blood oxygenation. So if you're looking at an image here, the corresponding brain activity for that is not until you get to like here, right? And so there's temporal autocorrelation. There's like, there's this very noisy decaying signal across the images that are being viewed. And that does affect how you want to do things like shuffling the data sets, how you go about, training things. It's a lot harder to predict on a held out scanning session compared to predicting the immediate next image or like the image behind another image of the same session that you're training on. In fact, like if you have a full session of scanning and you just remove the one sample, right? You could like recover that sample just because the signal from that sample has been blurrily contaminated into the next few samples in the series, right? If that makes sense, because it takes like 12 seconds to totally decay the signal for something that you perceive. So, knowing that does affect things like shuffling the data, processing the data, creating like the web data sets that we used was something that I was involved in that was useful, I think, to have some background in fMRI, and just understanding how noisy fMRI data in general is pretty important too.
Just because we have a little bit of time, and can you go back to the DALL-E 2 PyTorch page? Because I kind of stepped out for a bit. I put a thing in the chat, and missed a bit of that. Okay, so it's for the disjoint embeddings on there. Because I was thinking, it's like, why don't you just use like another open source model? But yeah. Thank you.
Did you have any ideas for other ways you could get around this sort of disjointed embedding problem?
Not off the top of my head, but this does seem to be like a nice area of research to go down.
Yeah, I think I forget off the top of my head. There were some other ideas that were discussed for this that we didn't try, but yeah, the diffusion model in particular is something that worked well for us, literally just copying the code or modifying the code. There were some things that we had to modify to get this to work with the very large MLP outputs. So one thing actually I can talk about this is that, so we initially mapped to the last layer of clip, right? Instead of the last hidden layer, because the last layer of clip is something that gets fed into the image variations model, whereas the last hidden layer is what's fed into the versatile diffusion model. And because we were mapping to the last layer of clip, we were able to use the pre-trained checkpoint from this GitHub. So this GitHub had already trained a text-to-image prior using clip. And so we took that, it was trained on Lion aesthetics, and we used that as the starting point to fine tune the diffusion prior to work for fMRI. Yeah. And that did way better to use a pre-trained checkpoint. And we had no checkpoint when we then switched over to using versatile diffusion, because the checkpoint was trained for the last layer, not the last hidden layer. So we trained from scratch for the prior because we didn't have any alternative for that. But given the fact that it helped so much when we were trying with the mapping to the last layer of clip, the assumption is that if we had a checkpoint, that our reconstructions would be even better. So, that's another thing that could have been done to improve MindEye, is train a text to image on the last hidden layer of clip, use that as the starting point checkpoint for fine tuning the prior that then gets used.
Oh, that's cool. That is something, again, to investigate within that, but yeah, a lot to take in.
Yeah. I guess one more thing I'll mention is that if anybody is interested in volunteering, obviously, like, get in touch, join the Monday meetings. One thing that I did is in our MindEye GitHub, I created these issues, and these are kind of things that we wanted to do but didn't have the time to do. So in the future, the idea is that we will refine this pipeline, like a MindEye version 2, have a better setup, get even better reconstructions, and that can be another deliverable publication. And so these are a lot of different like goals to try out for improving MindEye. And you know, everything's open source. If anyone's interested, can try looking into these different issues for how to improve things.
Got it. That sounds great.
I'm also
Yeah, hello?
Yeah, I'm also kind of curious, like, did you look at the voxels at all and try to make some, did you glean any understanding of like the spatial organization of images in the brain, or was that not something that you looked at?
That is not something that we looked at, but that's a great mention. So I think there are some other papers that looked at that. Ursulik's paper from this year did do some specific investigations into the different like functional specializations. So for instance, you can train a model on maybe just the fusiform face area. This is involved in face perception, and you can see that the reconstructions are more like they're going to be more face specific, like they'll do really well for images that have faces. Or you can try to synthesize images that maximize the activation of different brain areas. You can look at the weights for different voxels and see how they're playing a role for different kinds of image categories. So that's all very interesting stuff to look at, basically exploring the functional specialization of different parts of the brain using the natural scenes data set. There's other papers too that have looked at this recently. And I think that there's been recent work. I think I tweeted about this, but I forget the name of the paper, but showing new functionally specific areas of ventral cortex, like different, I think it was like hippocampal place area specific functional specializations or something like that. And you can do that by doing that kind of approach that I mentioned, which is kind of like a new exploratory analysis for uncovering functional specialization, which is kind of not the way it's typically done, where you basically have a hypothesis, like this area represents faces more than anything else. And so you present faces, and then you present all sorts of everything else, and you show that it had the highest activation for faces and not for anything else. That would be like the traditional approach. But this is a more like exploratory approach of you have such a diversity of images that you can kind of post hoc look at what's being most receptive across all of the brain and like look at the images that it's most receptive for and realize what kind of categories, what is being the cause for the differences and why it's responding more so to these images and than other images on a voxel by voxel level.
Thanks for the nice presentation, Paul. So my question is, how did you make the choice of the generative model here? Why is it diffusion?
So the basic answer to that is that diffusion models have been doing the best for text to image generation. So probably it would do the best for using brain inputs, basically. But we didn't just make that assumption and go with it. Initially, actually, I was trying to use StyleGAN and use map to StyleGAN embeddings and use that to reconstruct. And that didn't make it into the paper, but the results were not good. And then also we used, then the mind reader paper came out and they used a GAN as well. They used Lafayette. And as you saw, Alex, who's one of the co-authors on the paper, made these reconstructions by mapping to Lafayette. And you can see that it's not quite as good quality. So just pragmatically speaking, the results were not as good when we tried other models, but we only tried StyleGAN XL, Lafayette, Stable Diffusion image variations. We also tried unclip, and that didn't do as well. So versatile diffusion by far was working the best.
Okay. Okay, thanks.
I think it will, as new image variations models come out, you can basically see how high quality fidelity can you get if you take an image, convert it to the embedding that the model uses, feed it through, and then compare that output with the original. And the better that the model can do on that, I think the better suited it would be to being used for brain reconstructions. So like that's what does the best for versatile diffusion. When a new model comes out that does better with image variation, it should just be kind of a plug and play kind of thing, mapped to whatever space it uses. And assuming it does better at that task, I believe the reconstructions would automatically get better.
I see. Okay, thanks.
Yeah, and I'm not sure if there's any other models that anyone thinks of that could be swapped out for this. Like, if you have any ideas of other potentially good models to use as the reconstruction, I'm all ears to hearing what might work.
I don't know. You might have the best high point view of this, so yeah.
I'm looking forward to like Stable Diffusion XL or some new models coming out that we can use.
Got it.
Yeah, so I guess that's the end of this, unless anyone has other questions. But thanks for coming in and listening to me talk about the paper. And hopefully we'll have more journal clubs in the future. They will not just be like projects that we're personally working on, but we can talk about papers that have come out. And specifically in 2 weeks, we have a presentation from Arya Wang that I'll announce soon. But other than that, we don't have anything on the agenda. So if anybody is interested in just talking about a paper that seems relevant in some way to neuroimaging, then please either message me or send a message in fMRI General. And then we can schedule some journal clubs. It doesn't have to be like a formal presentation, but reading it in detail beforehand and guiding us through it would be very appreciated. Or if you just have like papers that you're interested in, we have an Excel, a Google Sheets page that you can find in the fMRI General channel that has some relevant papers if you're looking for a paper, or if you have a paper to suggest, then I would recommend checking out this spreadsheet. All right, thanks everyone. Hope it was you had a good
So
time.
is it
See ya.
sorry, is it a weekly journal club or bi-weekly?
Well, we have nothing scheduled for next week, so I mean, if there were people interested in presenting, it would be weekly, but if nobody is gonna help out with presenting, it might need to be bi-weekly.
Okay. Okay, thanks.
Yeah.
Just had a question, if that's okay. I was wondering if you tried experimenting with other imaging modalities like EEG or fMRIs. Obviously, like the spatial resolution is going to be a lot lower, but I'm curious, like, what types of images you can get.
Yeah, so I know that there's been some people who've been trying to do EEG reconstructions, and that seems really cool, but the data quality is obviously not going to be as good as fMRI. So in terms of non-invasive neuroimaging modalities, fMRI is going to give you the highest signal for for the task of doing reconstructions. However, that doesn't mean that you can't try other modalities as well. Something I'm particularly interested in is ECOG. So if you have like electrode data, then assumedly you're going to have a lot higher quality data to work with. So there is someone in the lab that I'm in who has been working with ECOG data and tried to do some reconstructions, but they didn't really have the time to fully analyze that. So maybe I can get in touch with them and try and work out a deal of using that similar data to try and do reconstructions. Yeah, does that answer the question?
Yeah. Is there a data set for ECOG that they were collecting? Or?
This is a private data set that's from the lab. So I would have to get permission to be able to share that and use it with the group.
Okay.
But yeah, like there's new data sets coming out. One really interesting avenue might be to, I'm not aware of any EEG data set of just like looking at images. I bet there might be something out there. Maybe it's even movies or something. But you know, you could think about multimodal applications. Like you can try to get the best of both worlds, essentially. Like EEG is really good temporal resolution, fMRI is really good spatial resolution. If you have data where you can essentially add multiple modalities to like clip space or any sort of multimodal space, that could be very promising. So I could imagine like image, text, fMRI, EEG, MEG, ECOG, and you have like all these data sets where you have some sort of paired data, similar to like ImageBind, right? Like they had everything gets linked into the image embedding, but you have different other embeddings like text and video and thermal and depth, whatever it was. You could probably do a similar thing with different neuroimaging modalities and hook everything into the image embedding space. That would be really cool.
Another question was, oh, sorry, go ahead.
No, go ahead. Sorry.
Oh, no, what you said makes sense. I'm wondering, so it seems like the data is really about like how you perceive images, but what we're evaluating on is like how well you can copy an image. So it's not quite the same thing. I'm wondering if you've like thought about other ways of training the model or other loss functions that might emphasize more the perception part of it.
That's a really good point. I don't know how you would, I guess you could do stuff like eye tracking. So like a lot of the times if you're doing fMRI, there's an eye tracker and you could get the data for that and essentially use that to inform like where are they paying attention to. And that could be useful in like, you know, if they're maybe paying attention to the person on the right side of the image, then the expectation is that you want to have really good quality reconstruction for that part that they're attending to. And it makes sense if the rest of the image is going to be a bit fuzzier and not have as much signal for. Maybe something like that could be useful, but I imagine it might be kind of hard to like quantitatively come up with a way to do that. Do you have any ideas for ways to do loss other than just using the original image?
Yeah, I'm not sure. I'm not sure. Yeah, I'm gonna think there.
Yeah, I mean, it is kind of like a not the exact best way to go about things because you're training the model you're training the model where the objective is you want the reconstruction to be exactly the same as the original image. But as I talked about at the beginning, our actual objective is not to do that, right? Our actual objective is to query the internal representation, like biased by your attention, biased by your personal experiences, and how you're actually perceiving the thing. But yeah, I don't really know how you would use a different loss function to address that difference in the alignment.
I think it's a bit of a sci-fi question. I'm sorry, but since it's measuring our perception, not the actual image, but what we perceive. So it brings in past experiences and noise. It is something that cannot be used, at least the current version cannot be, it's not intended for like criminal work or where people try to recall something.
It would be useful for trying to, like, one of the applications that I want to eventually do, my lab, the lab that I'm in is a computational memory lab. So one of the big things that we want to eventually do is reconstruct memories. So the first step in reconstructing memories is to reconstruct perception, right? So now we've kind of got into that. So the next thing we want to do is make it work for new subjects, like generalize across people. But after that, it would be really useful for being able to track memories. So for instance, look at something, encode it into long-term memory, and then maybe later on, I ask you to retrieve that memory, and then we reconstruct it. And a visual memory, that is like a photograph. And the idea is that you could be reconstructing memories in like fine-grained detail rather than the current method, which is kind of like a linear support vector machine of like how well does the patterns of activation look similar to or correlate with the original perceived activation. But if you can get like a fine-grained reconstructed image of that memory, like you can like diagnose like what's going on, what exactly is being degraded in someone's memory. Is it distorted in a systematic way? Is the context of what happened between encoding and retrieval changing the representation of that memory? Can you track a memory across time without the person explicitly retrieving it themselves? Like, is it possible to get a sort of like what would it be, like a tracking of an image across time? So like you have their representation and you can kind of gauge to what degree that representation is being activated across time. Like there are a lot of cool possible future applications for memory specifically. For criminal investigation stuff, yeah, I don't think that's not really something I think would be useful in the short or long term right now. Like the technology is not really going to be useful for that kind of applications. There's like some discussion of people have already tried to do lie detectors with fMRI, and it's just not going to be useful because of how easy it is to get around the system.
Right.
So, like even if we ignore the fact that you need a lot of training data, like if you move more than a few millimeters, the data quality is like totally noised and you can't really do anything with it. If you don't follow the instructions that the experimenter gives you, then the data is not going to work. So like you can't, there's a necessary aspect of participant compliance that's just a byproduct of neuroimaging methods being very finicky and noisy to use and easily messed up if you're not totally like trying to benefit the researcher themselves.
Right. I have a question about the fMRI general projects. What kind of general skill sets are you looking for somebody to start collaborating?
Anybody who has the basically coding experience, if you can just like help out with PyTorch models and can you work with Python and contribute in some way. Just people who have knowledge in general of like staying afloat of the literature of machine learning papers. Like I'm horrible when it comes to knowledge of the field of like, what is mix-up contrastive models? What are these different loss functions? What's the best way to train these models? What are the new embedding spaces? Like contributing ideas, contributing code, anything like that is valuable. And I think it's really worthwhile to just like talk about different things, of different approaches. And if you have an idea for a new approach, just do it, implement it. Even if you don't use it, even if we don't use it in the final paper, we still add you as a co-author. Like a lot of the co-authors in this paper, we didn't actually use the contributions, but I value the time and the investigations that were put into doing it. So like, that's still obviously very valuable. Like sometimes you try to do something and it just doesn't work as well as another approach, but you needed to do it in the first place to see how well it did. So yeah, ideas, code, fMRI knowledge in general too. Like, I think that's the benefit of being in an online community. People have different skill sets, and
Right.
yeah.
Yeah, this is very interesting. Yeah. I don't have a particular background in fMRI, but kind of started to gain some of it through Lindquist courses, which are really cool. Yeah.
What courses, sorry?
So on Coursera, there's a professor from Johns Hopkins, Lindquist. Yeah.
And was that useful? I'm always looking for better resources to direct to people.
Yeah, I think I didn't have any background about fMRI. I was just interested, and they have a specialization where there are two very good courses in the same specialization about the principles.
Okay, cool. Thanks for sharing. Yeah, so any other things we can talk about, or should I wrap up?
I had one last thing. Could you mention a bit how the pre-processing was done on the fMRI data?
Yeah, so basically the test set was all of the images that were shared across all the subjects. So not all the subjects saw the same images, but all the subjects saw some of the same images. So that kind of is the standard test set that's been used across all these papers. And every image was seen 3 times. So you have like 15,000 voxels activations for every one of those images that gets averaged to create
So
one
you
sample.
average across all the time steps while they were looking at the image, or what was?
So every 2 seconds, an entire brain volume of fMRI is collected. So if you have a 3-second image presentation, you get a single brain volume. So it's not really a
I
time
see.
series, essentially. I mean, it is a time series, but it's not like you get multiple instances with a single presentation. So the pre-processing specifically was done by the natural scenes dataset authors themselves. They used a package called VLM single, which is kind of like the most fancy way of fitting a general linear model to the time series data in order to get single trial signal beta weights, which is the actual like stuff that we use to feed into the model to train the model. So specifically, like if we're getting into the nitty-gritty details, I think they used fMRI prep, which is another package that takes the raw brain volumes and does stuff like motion correction, distortion correction, magnet field distortion correction, all this stuff to get the brain volumes into a good like usable state, like free of like artifacts and stuff. And then you fit a general linear model on top of the outputs of that. In their case, they used GLM single. And that fits like the hemodynamic response function and everything, deconvolves overlapping hemodynamic response functions on top of each other in order to get beta weights for every TR or every 2-second sample from the machine. And so that beta weight per image is what the actual sample is. And so then the beta weight for every voxel of that volume. So then like when I say brain activity, I'm actually referring to the beta weights of the general linear model outputs. And then averaging across the 3 samples for the same image repetitions. And like that's what gets used in the test data set.
Gotcha, thanks.
Yeah, we were very lucky that we didn't actually even need to do all the pre-processing ourselves. The National Scenes dataset authors gave us the, state-of-the-art quality pre-processed beta weight outputs for different brain regions.
And how long does it take to train the entire thing?
So it takes around 8 hours on a single A100 GPU for a single model of the high level pipeline. So the diffusion prior plus MLP gets trained at the same time end to end. So that all fits on a single GPU around 8 hours. And then separately, the low level model gets trained. I think that takes like 12 hours or something. And then on top of that, it takes a long time to then just evaluate everything through the generate the diffusion outputs of the final model. But yeah, the actual model for the main pipeline is about 8 hours. So not that bad in my opinion. Like that's quick enough to iterate on. Okay. Yeah, thanks. Amazing questions, by the way. Like I didn't expect to have such good discussion. So very happy that all of you are here. And yeah, thanks everyone. And see you around. Take care.
Thank you.
Thanks.